We don't have nearly enough time to cover the field of gene therapy like it should, but we'll do our best. And we have just a terrific group of panelists here to help us appreciate where gene therapy is headed. I'm Joshua Schimmer. I'm one of the Piper Jaffray biotech analysts. Have really been delighted to see the explosion of interest in gene therapy over the past 2 years. It is such an important year for the field as well. We're going to get multiple data readouts in ophthalmology and heart failure, spinal muscular atrophy, more data in hemophilia, and to help us hopefully see how the field may evolve in the coming years. As I said, just a terrific group of panel members. So maybe we'll start with introductions to my immediate left, which would be your right. Richard Lund, consulting professor at Cardiovascular Institute at Stanford University and consulting scientist at Cardiogen Sciences. Next to Richard, we have Ken Mills, the president and CEO of Regenexx Biosciences. Next to Ken is David Kirn, the CEO and co-founder of 4D Molecular Therapeutics. And next to David, we have Stephen Paul, the CEO of Voyager Therapeutics. So really amazing stories and companies emerging. All private companies at the current time, but really some innovators. Maybe to start, and Steve, why don't we start at your end and work back. Just tell us about Voyager, the history, founding, and the mission, and what you have going on.
Well, Voyager Therapeutics is an AAV gene therapy company focused primarily on the CNS, the central nervous system, the brain, and the spinal cord, and really focused on very serious disorders, things like Parkinson's disease, late-stage Parkinson's disease, ALS, amyotrophic lateral sclerosis, Huntington's disease, Friedreich's ataxia, diseases where the genetics are very clear. We know kind of what we need to do to treat the disease. Can we effectively deliver a gene to the right cell in the right tissue? Can we knock down a gene in the right cell in the right tissue? And that's really what we're doing. We're very broad, though. We're not just focused on these, what we call our product platform, but a lot on our product engine. So we're very into capsid engineering, new vectors. We believe it's still very early days with respect to these viral vectors being drug carriers or drug delivery machines, if you will. So we're into capsid engineering for our purposes, delivering to the brain and other tissues, et cetera. We're into manufacturing, very unusual for a startup company, but if you want to produce these vectors at scale, you need a process to produce them. So we've recruited some people who've literally invented the baculovirus SF9 process for producing AAV vector at scale, and we've set that up in-house. We're working with Commonwealth of Massachusetts on actually setting up a manufacturing facility, which is going very well. And then, of course, we're very interested in delivery. We have to get the vector, the gene, to the right tissue. And for the CNS, we're focused primarily on the spinal cord because we can do this relatively non-invasively through intrathecal administration or directly in our Parkinson's program, directly into the part of the brain that's deficient in dopamine to get that dopamine level up to treat the disease. So that's the main focus of Voyager. It's a typical third rock company. We spent a couple years in incubation, brought in some terrific founders, Chris Bankowitz from UCSF, Guangping Gao, who did many of the Regenexx vectors when he was in Jim Wilson's lab, who's now at UMass. Phil Zamore, who's an RNA biologist, one of the founders of Alnylam, just terrific. And Mark Kay, who's a very well-known gene therapy guy at Stanford. And they provide not just, inspirational leadership for the company, they actually are working with us on various projects. So that's in brief the Voyager story.
David, we're going to skip you, actually, and come back to you. But just kind of in terms of the flow, Ken, because Regenexx and Voyager, there's a little bit of a connection here. Why don't you give us Regenexx Bio's position in gene therapy and some of the relationships you have, as well as some of the talent.
Just quickly, we did announce, and I need to mention this, a very important collaboration with Genzyme Sanofi a few weeks ago. Terrific group of scientists that have been in the field of gene therapy for a number of years. For us, a kind of a transformational deal, brought together the two companies in a big way, and that's off to a great start.
Great. Ken?
Yeah, thanks. So I'm Ken Mills, co-founder and president and CEO of REGENXBIO, which is also an AAV gene therapy company. that was started by licensing in exclusively technology that was created in the lab of Jim Wilson at the University of Pennsylvania, and was based on discoveries of novel, naturally occurring adeno-associated viruses that had properties and characteristics that were dramatic improvements to other naturally isolated AAVs that had existed. This happened in the early 2000s. We formed REGENXBIO in 2009 because we saw a composite image of a lot of interesting translational and preclinical work going on in labs throughout academia and starting to occur in some early stage companies that we thought could be real products. And so we created REGENXBIO with the concept that we were going to be an enabler and enable people to move programs forward with this really important and special technology. The original technology had actually been discovered under about an 8-year sponsored research program from GlaxoSmithKline that really afforded Jim and Guangping and colleagues in his lab the ability to think about research in the discovery and advancement of AAV technology, I think, in a very unique and special way. And its yield and benefits have been significant. These vectors have gone on and are known now to have established really important clinical human proof of concept in several diseases, including hemophilia, a New England Journal of Medicine article that I think was a real turning point for the AAV industry at the end of 2011, and certainly from our perspective as a small company attracted a lot of new investment and support from large pharma, including groups like Genzyme Sanofi. We've evolved our model more recently to continue to enable through licenses companies like Voyager, and we actually have a network of 8 licensees who are at various stages of development. Several of them are in the clinic with our novel technology. But more recently, at the beginning of this year, we also took investment directly into REGENXBIO to advance our own programs using this technology in neurodegenerative disorders and retinal diseases. So we're a platform company first. We're evolving into a product-focused company. We think the benefits of the technology have proven themselves, that there are real products in this generation of technology. We're excited to be able to kind of advance those contributions. Several small companies, including Voyager, Avexis, AV Life, and a company we formed in the beginning or late 2013 called Dimension, are advancing those programs, and new clinical studies will be starting this year in early 2016. There's other companies that we're talking to that maybe people haven't heard of that will probably form this year and will become licensees of REGENXBIO as well. As that network expands, as our own programs move forward, we think it's having a major positive impact on the advancement of unique and novel treatments for patients.
Great. David, let's come back to you and tell us what's going on at 4D.
Perfect. So thanks for having me. I'm Dave Kirn. I'm the co-founder and CEO of 4D Molecular Therapeutics. So our company is an AAV gene therapy company. We have 3 areas of, we believe, differentiation and value creation in the field. First of all, we have a very robust vector discovery engine that can identify targeted and customized and highly proprietary vectors for any tissue in the body that we want to target. Number 2 is we have deep vector development experience. So our team has developed over 10 different vectors, over 30 different clinical trials, over 800 patients treated with viral vectors in the clinic. So I think that deep expertise will allow us to develop our own products. And then I think thirdly, we have a diversified business model where we will have our own products, but we'll also partner with complementary companies in specific disease areas. We do have partnerships in place today with Roche, Unicure, and Benetech, and we're in discussions with a number of other parties, including some of this in this room. So we're excited to develop our own products, but we're also excited to work with our partners to develop those products. Briefly, our discovery engine is called Directed Evolution. It was developed by my co-founder, David Schaefer at UC Berkeley over the last 15 years. He's generated a library of over 100 million variants using over 20 different methodologies to instill diversity into the library. And with that starting point, we then can do iterative selections for target tissues in non-human primates and with human tissue to identify vectors targeting specific tissues in the body. I think this should not only give us better products for the existing indications, but also open up new markets that can't be addressed with the current vectors.
Richard, why don't we come to you, and if you could share your background. You've had an important role in the development of Factor VIII Therapeutics, and now involved, I think, in some of the cardiology gene therapy approaches. Maybe just kind of take us through some of that history.
Great. Okay. Thank you, Joshua, and thanks for having me here. Yeah, a little bit briefly about why I'm here by accident. I did my postdoc to try to study eukaryotic gene control, and the means to an end was to clone the first human genes, which hadn't been cloned at that point, because my advisor, Tom Matiotis, thought the fetal to adult globin gene switch would be a great system for studying eukaryotic gene control. And little did we know that making the human genome library and cloning those genes was going to lead to things off the path from our grant proposal that turned out to be far more important. Because of that, I wound up probably at Genentech as a senior scientist for 11 years. And we were agnostic to diseases and programs, and we switched projects every year or so. Coincidentally, now that I'm in the gene therapy world, one of the genes I cloned was lipoprotein lipase, which happens to be Glybera, the only so far approved gene therapy product. And again, to tell you a little bit about accidents, I cloned it because I met someone at a meeting who said that this could be a diet treatment to dissolve triglycerides. And so we cloned it. And Genentech decided people were not going to take injections to lose weight, and that was the end of that project, and we moved on. Probably the most fruitful project I moved on to was Factor VIII for hemophilia, which was a wonderful run, and we created one of the two sold now recombinant products at a time when we were told every hemophiliac was hepatitis B positive, and what we didn't learn until for another couple of years was every severe hemophiliac was HIV positive at the time. And I think a little bit of a parallel to what the gene therapy field is going through now, the first projects were the low-hanging fruit. They were monogenic disorders where you had a protein that would treat the disease, but it was either impure or tough to purify from a human or animal source. And it was rather obvious to go after things like insulin and growth hormone and Factor VIII, as it is now in the transition to gene therapy to go after things like hemophilia or things that are untreatable. So Cardiogen Sciences. I was at Genentech for 11 years, then I went to Stanford, the Department of Medicine and CV Therapeutics. And learned about a year ago that Lu Lang, who was CEO at CV Therapeutics, was putting together a new co to do gene therapy for inherited cardiac arrhythmias. So that's what Cardiogen Sciences is focusing on. Silvia Priori in Milano is the scientific founder who first identified the defect in the calcium binding protein that causes a very bad arrhythmia that's fatal in kids who get stimulated and keel over. And it's a small gene, so it fits into AAV very nicely. It's a good first project. It's a recessive disease. It's called CPVT for those who care. And Dr. Priori created the mouse knock-in model, then made the AAV vector containing the wild-type gene and essentially cured the disease in a mouse model. That will be the first project at the company that we're going to move on to large animal farm tox and hopefully the clinic in a reasonable amount of time. But I'm not interested, and we're not interested in being a one-shop wonder. So we would like to pick the next several cardiac arrhythmias to go after. They're going to be more challenging, which is why I've been talking to some of these people already, because they'll either be dominant diseases where you have to either silence the aberrant allele and or also express perhaps more of the wild-type allele. They may have larger inserts. So they're going to move towards the next generation of AAV gene therapy vectors. So I want to be involved in that. I think it's really exciting. Some of you who may or may not have wasted your company's money investing in gene therapy 15 years ago, it was premature then, but now the time is right. You all know that. The field is exploding, and it's exploding not because it's a new century, but because the scientists have come along and created lentiviral vectors and AAV vectors that are much safer and more efficacious than the first generation of gene therapy attempts. So I want to be part of that, and hopefully Cardiogen Sciences will be involved in some of those targets.
Well, I love the title of the panel, gene therapy delivery, what can be accomplished with existing vector technology, how should we expect it to evolve? I think it's really such an important question to think about as we anticipate the twists and turns we might see for the field. Steve, let's come back to you. How do you know you've got the right vector for the diseases that you're interested in pursuing?
Yeah, that's a great question, Josh. I mean, it's a little bit like drug discovery in general. How do you know you got the best drug to take to the market? Is it good enough? And that's often the problem or the question on the table, because as time goes on, you might be able to improve even further. We believe that the current capsids, the current vectors that we have are good enough for our purposes to deliver genes safely to the brain or to the spinal cord. Now that by no means means they're the best, but they're good enough. We know we can transduce in a large species, a monkey, for example, non-human primate, virtually all of the motor neurons. Certainly well over 50% of the motor neurons up and down the spinal cord from the cervical region down to the sacrum. And that for treating something like ALS or dorsal horn DRGs for treating Friedreich's ataxia should work. And so those are vectors that are here and now. Now we want to know which ones among the ones that we can choose among are actually the best for that purpose, and both in delivery, sort of efficacy, but also in terms of safety. But it's my belief, as good as the vectors are that we have now, that we haven't really begun very much work. I mean, there's some wonderful work from 4D, and we believe now at Voyager to actually not only phenotypically screen thousands and thousands, if not millions of variants, but to sort of look at which ones are emerging as being better. What are the features of the capsids that allow you to get more into the brain or more into the spinal cord in our case, or more into the liver, or more into the heart, or more into the lung? And then I think we can do a somewhat more traditional sort of rational design, structure-activity relationships to really optimize the capsids. And that's where I think the field is. We're just beginning. There's other things beyond safety, immunogenicity, and can you dial in and dial out the kinds of properties that you want in these capsids. So my contention is that 3-4 years from now, we'll have even better vectors than that are currently here. But of course, I share, I think, the belief of our panelists that I think we're ready to go now with the ones that we have.
Dave, we're going to skip you again.
Wow, that's tough, man.
But we're going to come back to you
follow,
again.
so that's good.
Yeah, nothing personal.
And because you have such history and breadth in the field as you look out, do you think everyone's got the right vector? I'm not going to, if you think someone doesn't, I'm not going to ask you to highlight them, but if you think someone really does have it, who do you look at and say, those guys, their vector and their disease really line up nicely?
Yeah, I think everyone here can agree that we've hit a new threshold and a foundation for AAV gene therapy in particular that I think suggests that we are going to see products evolve from existing technology, and I think that's really exciting. It's clearly attracted a lot of investment, a lot of support, and a lot of great thinking and science. I mean, to sit here with these scientists as an example. I mean, when we started Regenexx several years ago, I'm not sure we would have gotten the pedigree of scientists in a panel discussing AAV gene therapy that we can today. And that speaks meaningfully to me to two things, Josh. One is that people see products here. There are treatments, and we're seeing not just great scientists, but clinical development teams and commercialization teams as it relates to manufacturing coming into AAV gene therapy to impact that meaningfully in a lot of companies. And a lot of those companies are getting financing from yourselves and from supporters and partners. And I feel like that was led, honestly, by the transition from vectors that were known in the early 2000s to the discovery of these novel isolates that Jim identified in the early 2000s. But it certainly doesn't stop there, and I emphasize and support that with more research and more investment, things are just going to keep getting better. And that's the natural evolution. I mean, Dick, talking about protein therapeutics and monoclonal antibody development, I mean, the product engines are going to get more sophisticated, especially as commercialization occurs. Then really the floodgates are going to be able to open for research and development in a way that I think is going to advance this. But the message is, it's here to stay. I think we're going to see improvements in the next 3 to 4 years. To echo what Steve said, I think we're going to see products in the next 3 to 4 years as well, and I think people should get excited about that. It's likely to be the low-hanging fruit, but the low-hanging fruit is really important because I think it's in diseases with high unmet need, where literally there may be no treatments available today, or where there is a significant improvement where we could benefit from a cost perspective, from a caregiver-provider perspective. And at REGENXBIO, we have 8 licensees that are working on 15 different programs with just the fundamental technology, all advancing products. I think we'll have 7 companies in the clinic next year of the 8 across different diseases. And so, we've seen this paradigm evolve from 2009, 2011, 2013, and it's significant. I think that as we keep building and keeping improving on that, the AAV field and gene therapy in general is likely to become as large, if not larger, than the biologics industry today. And I think that's something that the major turning point was, kind of the early 2000s and evolving to today and bringing in more science.
And you have a group of vectors identified. How do you see the role of generating new vectors and potentially improving upon the ones you have?
Yeah, I mean, fundamentally for us at REGENXBIO, since we have access exclusively to the platform, when we think about our programs, I mean, we start with what's available, and we think we have a best-in-class foundation. But I agree very much, with what Steve said, that there's areas where there still needs to be improvement for the class of AAV treatments, period. And I would say that immunogenicity is a great example of that. These are things that are foreign to the body. These aren't, humanized monoclonals or protein therapeutics, even if they're made in non-human or other forms of cells. These are viruses. And even though AAVs in particular are non-infectious or thought to be non-pathogenic, it doesn't mean they're not immunogenic in some form. Even in the clinical studies, I think we're seeing some evidence of that, which seems to be treatable and manageable in several of the diseases where AAVs have evolved and shown really good safety and efficacy. But I think if we want to get to that bigger bolus of diseases where this can really have an impact, there's going to be improvements that need to be made there. I'm a little, personally, and I'm the least qualified of the scientists on this panel to say this, but I'm a little conservative about my thinking about how much improvement in AAVs is available to drive more protein expression. I think that better routes of administration, like things that Voyager is exploring in terms of how to approach the brain, or maybe what Cardiogen might do about how to use AAV in the heart, is actually going to have meaningful improvement for efficiency of AAV. And I think immunogenicity and improving responses to the immunogenicity of AAVs will have some improvement. I'll be surprised if we see something in the AAV space that comes along that expresses 100 times greater than what exists right now.
Richard, why don't we come to you, and as you think about Ken's comments and applying them to cardiology, how do you think about delivery? How do you choose the right vector if you've got the target that you've identified?
Right. Well, I think in terms of the cardiovascular targets that we're looking at, the way has been paved by the projects that you all know about and have heard about some of today, with AAV delivery for heart failure or post-MI, using catheters, which, as somebody said this morning in one of the breakout sessions, cardiologists are used to doing this, and in 20 minutes, they're used to delivering something into the heart. They might as well be delivering AAV. So I think general cardiac cath delivery into the heart is going to take care of a lot of issues that would have been there in terms of dose of virus and getting some localized effect. I think what's always going to be important, but again, so far the data looks encouraging, that you're not going to get a lot of off-target expression, and if you do, it's going to be fairly benign. And when I was watching the gene therapy field from a side 10 or 15 years ago when I was not doing gene therapy, clearly one of the big problems then was vectors were integrating DNA randomly, or even if they were touted to be selective by homologous recombination, there was enough off-target integration to cause problems. And that was one of the problems. It was oncogenic potential with the first generation of vectors, because, as I like to say, it's tough to beat Darwin And even in a very rare event, if you got integration near a gene that either because you turned it off or turned it on, just sped up the cell cycle a little bit or inhibited apoptosis a little bit, that cell is going to take over and cause problems. That was one of the biggest things that had to be solved in gene therapy. I think that's been solved pretty much with this generation of vectors and deliveries, but one of the challenges to really expand gene therapy is going to be how to get the gene editing and gene correction mechanisms that we're also excited about that work in cells and culture, but so far as nobody can squirt this system into a body systemically like you can with the current AAV and get gene editing to work. That's going to be one of the great challenges. It's going to happen. It's not like traveling faster than the speed of light. It's not impossible, and I think it will happen. And that will greatly expand, as Ken was saying, the number of disease targets that we can tackle.
Dave, why don't we come back to you, and as you think about your objectives with evolving vectors, what are your target profile? What degree of increased protein expression are you hoping to achieve? How do you think about deploying your platform strategically across indications? And then, maybe as part of that, how do you also think about optimizing a vector in a preclinical setting, but then ultimately having that translate into the human setting as well?
Right, so those are great questions. So I think, first of all, again, the status of the field today, the reason we're all here and it's been so successful of late is because some very intelligent clinician scientists were able to identify the low-hanging fruit indications, the clinical proof of concept indications where existing vectors, which are natural, which didn't evolve to be products, weren't engineered as products, but were really lab contaminants or monkey infections, they found a way to brute force those into indications and get clinical benefit, which is a tremendous success. The routes of administration have had to be difficult, oftentimes subretinal or 70 injections into the skeletal muscle to get that benefit. So it's a huge success to be where we are, but I think we can do a lot better. What we're trying to do is identify and develop vectors that, number one, will give us much better infectivity, not only more viruses per cell, so more protein per cell, but a greater number of cells within the target tissue. We want to use clinically validated and routine administration methods. So for the eye, we'd like to go intravitreal rather than subretinal, for example. For skeletal muscle, we develop vectors that can just be given IV and target a wide range of muscles rather than intramuscular injections. So I think it's better efficiency, it's better delivery methods, it's evading pre-existing antibodies. Most of the existing vectors, half the population will be antibody positive, excluded from the trials and presumably excluded from treatment once it's on the market. So we need vectors that can evade antibodies, pre-existing antibodies. These, again, are things that the technology that we're developing has been shown to do. We have proof of concept we can achieve this. And then I think the other big thing is manufacturing productivity limits a lot of these current vectors with the existing manufacturing process. So one of the reasons the eye is attractive is because the doses are very, very low. But when you want to go IV and treat hemophilia, for example, you're going to have something that A, is very efficient, and B, manufactures very well. So I think that's another area where our vectors can improve upon what's out there. And then the last thing is if you have a more efficient vector that is effective at lower doses, you're going to have less likely immunogenicity, less likely to get transgene and vector clearance down the road. So I think those are the features that we want to build into our vectors. And again, we have proof of concept for each of these in isolation. Now we're in the midst of identifying vectors that have each of these features. And again, over the next 9 to 12 months, we'll have 12 different selections completed and have those vectors in hand.
Yes, depending on the duration of benefit from one gene therapy product, there may be a need to give the exact same product with the exact same profile with almost no differentiation, except it's a different vector. So you're the next line of therapy 5-6 years later. Is it too early to start strategizing around that because we don't really know how long the durability is going to be?
I think it's a great question. I think there's two aspects to that. Number one is just even today, most of these disease indications are going to exclude half the patients because they have pre-existing antibodies. So right there, you can come in with a fast-following product and just address those if you can evade the antibodies that are precluding those patients. And again, that's something we'll be doing. And then the other question is there going to be serial treatment, either because the gene effectiveness wears off over time, or even if you just get partial correction. There may be diseases in the eye, for example, where a patient can now ambulate through a maze, but do they have normal vision? Probably not. And so the question will be, can you come in on top of that with another vector that does not cross-react and get that sort of benefit?
But Josh, to your point, I think if you're working to deliver a gene to a tissue that proliferates and turns over, you do need to be mindful of the fact that that may not last forever. In the case of neurons, which is what we're going after, these are post-mitotic, they're terminally differentiated. For all intents and purposes, they'll last for the life of the organism unless they die. And so we're preventing them, trying to prevent them from dying. But if you're going after liver, how long will these hemophilia treatments last? I don't know. The heart, I'm not sure. But these are cells, since these are episomes, they're non-integrated, you're going to have to think about potentially redosing at some point down the road. So I think, and we do this, we think about this in the case of vaccines and things like that. So it's not out of the ordinary. There is some data suggesting that you can immunize animals to one serotype, very active immunization with adjuvant, and show no cross-reactivity to a more distant serotype. Or ideally, find capsids where there's no pre-existing antibodies that exist that will neutralize the vector from doing its job. So I think there's a lot of thinking. The other thing I would add is the capsids are very important, but let's not forget there's more to the payload. There's the promoter, there's codon optimization. Do you want your gene and your protein expressed in all cells in the body? You may not, and at what level, particularly if it's a protein that's not normally expressed in that cell. I don't know how nature will like it, but if it's expressed 3-4x what otherwise wouldn't be expressed in that cell, that could be an issue. So using specific promoters, you can engineer those vectors. In our case, we can use promoters that only express in neurons or only express in glia, and it seems, at least in the animal studies, to work very well.
Maybe on a question related to that point, it maybe to do with biomarkers or the lack of biomarkers, because if you're going to deliver gene therapy to the brain,
right.
I'm not sure there's a scan you can do that's going to light up all the areas of the brain that were successfully transfected. What's interesting in the application for hemophilia is that you've got a very obvious blood biomarker, some immunogenicity profile. You can kind of piece together maybe what's happening in the liver kind of circumferentially. How do you think about that in the CNS? And how does that also impact disease selection?
First of all, you can't say enough for having a useful biomarker for anything you're going after, any disease. At Lilly, towards the end of my tenure, we had literally biomarker programs on every program in clinical development. If not to check about target engagement, some pharmacodynamic marker that said you're going in the right direction. So it's very important for gene therapy too. In the case of our Parkinson's program, we're putting in the brain with a viral vector, with an AAV vector, a gene that encodes a protein that converts L-DOPA, which is the amino acid that patients take to treat Parkinson's disease, to dopamine. Turns out we put it in a very specific area of the brain, the area with the brain where dopamine is deficient in Parkinson's and where patients have to get more dopamine to control their motor symptoms. This is in late-stage patients. We can image that enzyme. We have a PET ligand, fluoro18 dopa, that we can give patients before the delivery and after delivery and see if we've done it, if we've got it. We can also infuse these patients with dopa itself. There's something called a dopa challenge test, which was developed many years ago. And we can challenge the patients before and after and measure motor symptoms, Parkinsonian motor symptoms, to see if we've done anything. And that is very, very vital to us being able to determine the right dose, the right coverage, and all those and sundry things. Now, it's not as easy for all of our programs. That one happens to work very well, but we're working very hard on other biomarkers for virtually everything we're going after.
Love to hear the other panelists' thoughts on that topic. David.
The use of biomarkers specifically?
Yeah,
well, I think,
how
I think,
important is it?
yeah, I think when you pick disease indications, it's going to be important to pick them wisely. So not only can you deliver using a clinically routine method of delivery, but can you identify short-term clinical endpoints, which are reproducibly measurable, clearly measurable, and where we have good validation that that correlates with eventual patient outcome. Because I think that's what makes a disease target very interesting, at least early on in the history of the field. So that's why hemophilia is so attractive. We know that blood levels correlate directly with outcome. It's easy to measure. It's reproducible. And I think so. Choosing indication we can do that is really important.
Yeah, Ken, your thoughts?
I think the standards, like Steve said at the beginning, the standards on the issue of biomarker are similar for gene therapy as they are for all drug development. And I would say, even we're developing programs for neurodegenerative diseases, and through discussions early on with Voyager, I think we share many of the same opinions that for our case, we're developing treatments for lysosomal storage diseases, and we need broad distribution in the brain, and we're looking for downstream biomarkers because we think we can get both intracellular activity with AAV, but also get cross-correction through the secretion of proteins that are translated intracellularly. And so we want to look for biomarkers both in the CSF, as if we were administering ERT to the CSF, and then we're also able, through natural history and aligning ourselves with really thoughtful KOLs, to get some imaging data too to help support that we're changing the brain morphology. So I think, but that comes from work that Biomarin and Shire and others have been doing to track these diseases for a while as well. So I think it's a great time for AAV to be entering into a space where big pharma and biotech has been thinking about these issues for a while. We benefit.
And Richard, when it comes to cardiac applications, what are the biomarkers or are there early measures of success that let you know
at first
you're
I
on
thought,
track?
well, the heart, the first thing you want to target, near to my heart, is hemophilia, because as David mentioned, but there are a couple of other aspects. That's really low-hanging fruit, even though there's an existing therapy. So, people can take the recombinant protein. Why do gene therapy? Well, there are reasons. Believe me, kids don't like their mother injecting them every 3 days with the protein, and some people convert to the gene therapy. And it's really low-hanging fruit because all you have to do is get some of this protein in the plasma. It can come from hepatocytes, it can come from other cells in the liver, it could come from cells in the muscle. We know it works just by showing up in the plasma. That's one. Number two, no one has ever overdosed on factor VIII or factor IX. And as a matter of fact, it's well known from the spread of clinical effects that if you get expression up to maybe 5% normal levels, you really improve the life of a hemophiliac. But another thing that wasn't mentioned is you don't have to control the expression. It's constitutive, and that's just fine. So I think one of the next generation of molecular biology contributions to gene therapy will be to figure out how you incorporate responsive promoters to things such as insulin or growth hormone, which are expressed in timely fashions depending on the state of the body at the moment. That'll happen too. We're not ready for that right now. In the heart, for these arrhythmias, the biomarker is whether or not the patient suffers from the arrhythmia. And in the first one, CPVT, that we are looking at right now, it's actually a routine test where there's a catecholaminergic stimulus that triggers the arrhythmia in the physician's office, and then it's withdrawn and they recover from it. So it's not as easy as doing a blood coagulation test on a little bit of blood, but it is a readily available biomarker. So we're okay with that. I think in the brain, you're going to solve more difficult problems in the future where you've got to get wiring right, and you may or may not have imaging biomarkers, but we all know people are trying to discover those for Alzheimer's and for your other targets. So I think a disease where there's absolutely no possible biomarker won't be touched for a long time to come.
Why don't we see if there are any questions in the audience back there?
If you address this, I'm sorry if I missed it, but we all want to believe that gene therapies can be one and done, and that we hopefully cure a patient. but can gene therapies be re-administered to a patient without fear of immunotoxicity?
Ken, why don't you?
Yeah, so I think that the answer to that is that not the same treatment that's given to a human one time can be re-administered. And I think, but a different delivery mechanism, there exists the possibility to retreat and continue to see that constitutive expression that was delivered even with the first treatment. And I know at Regenexx and through other of our licensees, we have evidence of that in many disease models, including non-human primate models. I think what you have to be careful about, and I think both David and Steve touched on it, is, what is the overlap, if you will, of those two vectors, the first one and the second one you're coming in with later, and you have to both screen populations and see who's going to be responsive to which one, and then think about that on re-administration. But I think it's possible. The other thing that I'll say just in general about durability is, I agree certainly that there's an unknown there, but there's a lot more data than there was 5 years ago. and we certainly have evidence now of gene therapy treatments that through a variety of readouts are showing expression levels that are consistent with what they were on day one out 4 years, I think, in hemophilia. We're coming on the 5th year of certain patients that were treated by a group at UCL. In some of the retinal diseases, RPE65 being a case, I think they're out 6 years or maybe even more from some of Gene Bennett and Jim Bainbridge's early work. So there's real, I think, excitement, but we're still learning. And I think in some diseases, that's not going to be the case. But hemophilia is an example of, I think people were transducing hepatocytes and we're expecting turnover. I don't think yet we're seeing as much as a lot of people thought a priori, certainly compared to brain or muscle. But I think, we're learning. N is small. I think, we have like 20 patients in hemophilia that have been dosed. So.
Thank you.
There may be immunoprivileged sites. So the eye, you may be able to re-inject. I think the LCA experience, they injected the other eye.
Yeah.
Is that just an eye thing, or might there be, do you think
retinal
CNS
neurons, are going to express for a long time unless they die.
Right.
And the same thing with just brain neurons. We know, for example, in our Parkinson's program, in monkeys, we can put the gene for ADC in the striatum, and 10 years later, the expression levels in monkeys are identical to what they were on day one using PET scans, and then, of course, we can actually measure the protein. In humans, we have data at least at 5 years in several patients where the pre and post PET scan look identical from the very first year PET scan, so that it's probably stable. And I agree that we just don't know yet exactly how long is long. If you're transducing 5% of hepatocytes and that's sufficient, what happens if that number gets to 1%?
Right.
I just think it's something to be thinking about, and I personally believe, given what I know of the literature, that you could engineer around that. You could actually give the same gene, but with a different vector, with a different capsid, and not have cross-reactive neutralizing antibodies that would preempt you from using that capsid.
I think there's a question here. Yeah.
Sorry, are there indications for which a non-vector approach is better from the get-go?
Well, I think yes. I think most of us are choosing indications where there's a pretty wide therapeutic index based on the gene that we're trying to deliver, where regulation is not going to be a big issue. You get above a certain threshold, too much isn't too bad. So, eventually we'll get regulatable promoters. We're not there yet, but right now I think we're trying to pick diseases where the benefit-risk ratio is very far in excess of the benefits of it. We're still, feel encouraged by the safety data that we've seen so far, but let's face it, it's still kind of early in humans and we don't know. We're all sitting looking what's going to happen with the current SMA trials that are dosing IV. Is that the liver's great and safe and whatever? Of course, this is a devastating fatal disease, so there, the benefit risk is, but we don't know a lot. So we want to pick diseases where there are no alternatives in our case, and where the safety bar is reasonably low given the alternative, which is no therapy, and the patients die. So,
Thank you.
Any other other questions. If not, maybe I'll throw out one more. Ken, it's going to be a very active year in gene therapy. What do you think we're going to learn this year?
So I think clinically and scientifically, there's at least a couple companies that I know in the public market space and even a few privately that are going to report data this year on some pretty compelling studies. And many of those are using what I consider to be first generation of technologies. So prior to Jim's discoveries that were the isolates discovered out of preps of adenovirus, where the name derives from. And those were adopted by companies that have been around for a while, and it was best in class at the time. So I think, and they have selected diseases that I think might meet the good enough profile. Not that there won't be improvements, but I think there's going to be this sense and I think expectation. I think of Celadon with the CIRCA-2A study. I think of Avalanche, which is going to report data on wet AMD. Some really large indications, yeah. And of course Spark, yeah, with the registration study for RPE65 that is going to tell a lot about the composite image of AAV. I don't consider it to be binary, Josh. I think it's going to evolve the story. My expectation is that I think there's going to be neutral to positive results coming out of those studies. There are a couple sleeper studies as new vector technology has emerged and as new drug discovery engines have evolved in companies like Voyager, and we're getting better and smarter about drug development. I think we're going to see better results through that combination. I think this SMA study that Steve was just alluding to is an interesting one to follow and may report data this year that could be compelling. And so it is exciting. I think, is this the year where it's a huge turning point? It opens up the gates for kind of, we know exactly what the solution is. Far from it, but there are huge steps to be made. And look, we, as companies and as drug developers, we're not going to be perfect. We're not going to have a 100% hit rate. There are going to be missteps, and we're going to step back and then I think take 3 steps forward. I think what's exciting is the amount of investment that's coming into AAV gene therapy this year and last year is meaningful enough to actually develop treatments all the way. I think the technology was there. Now we've got the people and the capital.
I think that's a great thought to end on, actually. So thanks to the panelists for a terrific discussion.